Cancer biology. Cancer is one of the most dreaded diseases of human beings and is a major cause of death all over the globe. More than 1 million Indians suffer from cancer and a large number of them die from it annually. Then now the question raises here, what is cancer? Cancer is basically a condition of failure of cell division control. Cancer is basically a condition of failure of cell division control. That means we say uncontrolled cell division. Uncontrolled cell division is called as cancer. To understand the cancer biology, let us see some terminology here. First of all, let's go to the neoplasm. What is neoplasm? Unchecked division of cells leads to the formation of neoplasm. In general terms, neoplasm can be called as tumor. Unchecked division of cells which leads to the formation of neoplasm. In general terms, we say it as a tumor. Such an abnormal proliferation of the cells is called as neoplasia. Abnormal proliferation of the cells of neoplasm are called as neoplasia. Formation of tumor. In general terms, we say formation of tumor. Neoplasm means tumor. Formation of tumor is called neoplasia. Yes, neoplasms or tumors are of 3 types. Number 1, benign tumors. Number 2, pre-malignant tumors. Number 3, malignant tumors. Benign tumors, pre-malignant tumors and malignant tumors. Let us see one after another. Number 1, benign tumors. Yes, benign tumor. As we said now, tumor means uncontrolled division of the cells. Here also you can see uncontrolled division of the cells here. But in general, these particular cells are going to form a fibrous capsular sheath surrounding them. And it is not going to spread from one place to the another place. Spreading of one place to another place, the feature we say metastasis. But you don't find such kind of metastasis here. So these are just a fibrous capsule. We see the uncontrolled division, but just a fibrous capsule. So such kind of tumors are very common here, but they are harmless. For example, if you take uterine fibroids in female human being is one of the example here. A small surgery itself can remove this particular tumor. So this cannot be called as a cancer. Benign tumors cannot be considered as cancers. Pre-malignant tumors. Pre-malignant tumors which are also considered as carcinoma in situ. Pre-malignant tumors which are considered as carcinoma in situ, in which cells transform into cancerous cells in due course of time. As there is no problem now, but malignant tumor nothing but called cancer. Carcinoma in situ or pre-malignant tumor, at present there is no problem, but they may proliferate and these cells can transform into cancerous cells in the due course of time. Let's see the malignant tumors here. These are the cancerous tumors. Malignant tumors are considered as the cancerous tumors. They have all the properties of the so-called cancer, that we say the malignant tumors of the body are called cancerous tumors. Let us see the properties of these malignant tumors or cancerous tumors. So for that we need to go with the comparison between a normal cell, the properties of the normal cell, then how it leads to a cancer. That losing the properties of normal cell itself, how it leads to the cancer here. So the reasons for the cancer, first of all, let us see a process. Generally cell are going to connect each other with the help of some adhering junctions. General body cells are going to connect each other like this. When they divide, in a controlled division procedure can be seen over. So these normal cells having a property called contact inhibition. Contact inhibition, which means cessation of cell division and cell mobility when they are in close physical contact with each other. Cessation of cell division and cell mobility. And that means as long as these particular cells are having a great bonding between them, that they don't show any uncontrolled division. That we say a property called contact inhibition. There is no possibility of uncontrolled division, that if any cell is trying to like that, we say cessation of cell division and cell mobility, then when they are in close physical contact with, as long as they have physical contact with each other. Then why cancer? Then question raises here, why cancer? Why cancer? Simply that failure of this property, that contact inhibition property. That means the cells are going to separate. The cells are able to separate each other. They don't have the contact inhibition property, and they divide rapidly, and they also can spread from one place to the another place. So failure of the contact inhibition leads to a property called metastasis. The failure of the contact inhibition leads to a property called metastasis. Let me once again, normal cells having, as long as they are having a physical contact in between them, that there is no possibility of uncontrolled division and there is no possibility of the cell mobility. That procedure in general called as contact inhibition. So then what happens in the cancer cell? They lose the property called contact inhibition. The failure of contact inhibition property leads to cell will, the mobility of the cell is possible. They move from one place to the another place, and that failure of the contact inhibition, moving from one place to another place and dividing rapidly, we say procedure is called metastasis. So that's why we said malignant tumors are having a property called metastasis. They can move from one place to the another place, and they spread the cancer. And in case of benign tumors, we said there is no metastasis. Metastasis is absent here. That's why they cannot move from one place to another place. Hence we say harmless tumor. Benign tumor is harmless tumor. Due to the property of metastasis only, we said that malignant tumors are cancerous tumors. Point to be remembered here. Generally, normal cell is having a property called contact inhibition. In a normal cell, contact inhibition, a property called contact inhibition, you see. But in the cancer cell, there is no contact inhibition. There is no physical contact. That's why a cell can move from one place to another place, and a cell can divide rapidly. The feature called metastasis. Now the question raises here, what makes a normal cell contact each other? What makes a normal cell, one cell to the another cell, how they bind each other? We have a special kind of protein called cadherin. Cadherins called calcium dependent transmembrane proteins. That normal cells are joined by intercellular adherens junctions called as cadherins. The cadherins present here, these cadherins which makes one cell to connect each other, which makes not to divide, uncontrollable division is not possible here, and metastasis is not possible here. So simply we can answer here how the cancer is possible. The absence of cadherins. The absence of cadherin. If there is no calcium dependent transmembrane protein, cells can't bind each other, and cells don't have control one on each other. That's why they divide rapidly, and they can spread rapidly from one place to another place. We said already the process called metastasis, and metastasis leads to the neoplasm cancer. Okay, so that to remember here, in a normal cell, cadherin present. Cadherin is there in normal cell. Absence of cadherin, the cells lose the physical contact. That adherens attachment is not possible here. That makes the cancer. The next, let's see the another property here, anchorage dependence. Anchorage dependence. What is anchorage dependence? Let's do a small experiment here in a nutrient medium. I am taking normal cells here. I am taking normal cells with providing a lot of nutrients which are required for the normal cells for their division. How do they grow? Generally, they grow like this. On the walls of that particular, they grow like this. In the mean period of time, you can see the cell division here. To the walls, these particular cells, we say, they grow to the walls. So this we can say simply, this is a normal cell property. Then what happens to the cancer cell? If you take the cancer cell, if you take a cancer cell in the nutrient medium in a mean period of time they divides rapidly, but you'll find that there is no attachment of these particular cells. Attachment of these particular cells to the walls you cannot see here. That because the cancer cells lose a property called anchorage dependence. Normal cell attach or anchor to the surface of the dish in which they grow when the medium is sterile and supply of nutrients is enough. Then now the question is, what happens when normal cells are grown in a dish containing all the requirements necessary? What happens? They grow rapidly, but they grow on the walls. What happens when cancer cells are grown in a dish containing all the requirements of necessary? What happens here? They grow rapidly, but they don't attach to the wall. So we can say with this experiment we can understand easily. Generally, this process we say biopsy. A small tissue of the patient can be taken here. If it tests like this, if the particular cut tissue of the patient is grown on the walls, we say he don't have cancer. But if that cut tissue of the collected from the patient, if they grow rapidly and they don't attach to the walls, simply we say they don't have anchorage dependence and this is cancer, malignant tumor. Another property of normal cell is apoptosis. Here we are going to find the reasons for the cancer. For that, first of all, we are observing what happens in general in normal cell. What happens in normal cell? If a normal cell is dividing rapidly, what happens? The cell recognizes itself the division is not going proper way. That's why they die themselves. We say the process is programmed cell death, apoptosis, programmed cell death. When in a normal cells, a cell division is going, uncontrollable cell division is growing. So that what happens to that particular cell realizes themselves and they die themselves. That process we say programmed cell death. Then why cancer? Simply we say failure of apoptosis, programmed cell death. Failure of apoptosis leads to the cancer. Failure of apoptosis. Generally, when a cell started uncontrolled division, when a cell is moving towards the cancer, what our immune system do? We have T cytotoxic cells which participate in the cell mediated immunity, and we have natural killer cells, separate natural killer cells. We learned earlier in the immunology, the T cytotoxic cells and natural killer cells are going to destroy that particular cancerous cells which are growing in the body. These particular cells are going to be destroyed over. Then now the question raises, why, how we get cancer? Simply we say failure of T cytotoxic cells and natural killer cells leads to the cancer. So let's learn about the gene P53. Gene P53 is a gene that codes for a protein called P53 protein that inhibits the development and growth of the P53 protein inhibits the development and growth of tumors. That means in any part of the body, if a cell started dividing rapidly, a cancerous cell is observed, the gene P53, we say it has tumor suppressor gene produced, a protein called P53 protein and suppress the action of that particular. Then now the question raises, how, why we get cancer? We have such kind of mechanism, how we get cancer? The two dominant genes, presence of two dominant genes of the P53 work efficiently. At least one dominant gene, they work efficiently. Simply we say in a person with cancer that mutation happened on the both genes, that both recessive genes if you observe in that particular person, that leads to the cancer. That means homozygous dominant person, heterozygous dominant person having two dominant genes, one at least a single dominant gene can makes the working efficiency of the gene 53 gene to produce the P53 protein to suppress the tumors is possible. But in case of double recessive, both mutation occur on the both genes, both became recessive, that we say in that person it may lead to the cancer, which they cannot work efficiently. The tumor suppressor gene cannot work efficiently. Gene P53 gene generally considered as a guardian angel of cell's genome. Guardian angel of the cell genome because it works efficiently and suppress the tumors by producing a protein called P53. But in those people, the failure of the two dominant genes having recessive genes may lead to the cancer. Similarly, we have a protein called a retinoblastoma protein is also a tumor suppressor protein. In case of human beings, we will observe this particular retinoblastoma protein is also a tumor suppressor protein that makes a dysfunctional in several major cancers. The function is similar to the P53 protein. Then now question raises, how we get cancer, the failure of these particular retinoblastoma protein. Then what we focus till the time, that what kind of defense mechanism we have in a normal cell till the time. So even we have such huge defense issues to protect from the cancer, how we get cancer? How we get cancer? Simply we said, whatever that defense activities our body is doing, if all they fail, that leads to the cancer. Now let's see different types of cancers here now. The first of all, carcinoma, types of cancers. Number one, carcinoma. What is carcinoma? Cancers of epithelial tissues or cells, which are most common as we say such kind of cancers as carcinomas. Epithelial cancer, we say 80% of cancers belongs to carcinoma itself. Cancers of connective tissues are called as sarcomas. Cancers of connective tissues are called sarcomas. Leukemias, cancers of bone marrow cells resulting in unrestrained production of the WBC. We say it as a liquid tumor. That leukemias are called liquid tumors here. Cancers of the lymphatic system are called as lymphomas. Cancers of the lymphatic system are called as lymphomas. Cancers to the epithelial tissues are called carcinomas. To the connective tissue called sarcoma. And liquid tumors, that uncontrolled production of the WBC, the cancer in the bone marrow, is called leukemia. And cancer to the lymphoid organs are called as lymphoma. Carcinogens. What are carcinogens? A carcinogen is a substance that causes cancer. Carcinogens damage the DNA. Several radioactive substances like gamma rays and some V rays, X rays, and inhaled asbestos, sudden dioxins, tobacco smoke, which contain many types of carcinogens such as benzopyrene, nitrosamines, and inorganic compounds such as chromium, cadmium, arsenic, polonium 210, are different the elements which are going to cause the cancer. We call them as carcinogenic. If the DNA damage is too severe to repair, the cells undergo apoptosis. What we said now, that particular apoptosis is failed, programmed cell death is failed, we get the cancer cell in this particular case, the various kinds of carcinogens are playing a vital role here to failure of our defense mechanisms. Whatever the defense mechanisms we said earlier, that all going to be done by carcinogens. Carcinogens are going to be major cause for the cancers here. Different kinds of cancers are caused by the carcinogens. Let's take a glance look now. The mechanisms that underlie the development of cancer, oncogenic transformation of the cells, its treatment and control have been some of the most intense areas of research in biology and medicine. In our body, cell growth and differentiation is highly controlled and regulated. In cancer cells, there is a breakdown of these regulatory mechanisms. Normal cells show a property called contact inhibition, by virtue of which contact with other cells inhibits their uncontrolled growth. As cancer cells appear to have lost this property, as a result of this, cancerous cells just continue to divide, giving rise to the masses of cells called tumors. As we said earlier, the tumors are of three types. Basically, in the NCERT text, it was given as two types, benign tumors and malignant tumors. What are benign tumors? Benign tumors normally remain confined to their original location and do not spread to other parts of the body and cause little damage. As we learn now, benign tumors normally remain confined to their original location and do not spread to other parts of the body and cause little damage. Malignant tumors. The malignant tumors, on the other hand, are a mass of proliferating cells called neoplastic or tumor cells. These cells grow very rapidly, invading and damaging the surrounding normal tissues. Metastasis, as we learn now, as these cells actively divide and grow, they also starve the normal cells by competing for vital nutrients. Cells locked from such tumors reach distant sites through the blood, and wherever they get lodged in the body, they start a new tumor there. This property is called metastasis, is the most feared property of malignant tumors. What are the causes of cancer? Transformation of the normal cells into cancerous neoplastic cells may be induced by physical, chemical or biological agents. These agents are called carcinogens. Ionizing radiations like X-rays and gamma rays and non-ionizing radiations like UV cause DNA damage, leading to the neoplastic transformation. The chemical carcinogens present in tobacco smoke have been identified as a major cause of lung cancer. Cancer-causing viruses are called oncogenic viruses, have genes called viral oncogenes. Study of cancers are called oncology. Furthermore, several genes called cellular oncogenes, C-onc or proto-oncogenes, have been identified in the normal cells, but which when activated under certain conditions could lead to oncogenic transformation of the cells. How do we detect cancer? Cancer detection and diagnosis. Early detection of the cancers is essential as it allows the disease to be treated successfully in many cases. Cancer detection is based on biopsy and histopathological studies of the tissue and blood and bone marrow tests for increased cell counts in the case of leukemias. What do we do in biopsy? In biopsy, a piece of the suspected tissue cut into the thin sections is stained and examined under microscope. Histopathological studies, we call this as, by a pathologist. Techniques like radiography, use of X-rays, computerized axial tomography, CT scan, magnetic resonance imaging, MRI, are also very useful to detect 157 types of cancers of our internal organs. The computed axial tomography or computed tomography uses X-rays to generate a three-dimensional image of the internals of an object. MRI uses strong magnetic fields and non-ionizing radiations to accurately detect pathological and physiological changes in the living tissue. Antibodies against cancer-specific antigens are also used for detection of certain cancers. Techniques of molecular biology can be applied to detect genes in individuals with inherited susceptibility to certain cancers. Identification of such genes which predispose an individual to certain cancers may be very helpful in prevention of cancer. Such individuals may be advised to avoid exposure to a particular carcinogens to which they are susceptible, like tobacco smoke in case of lung cancer. So identification of such genes which predispose an individual to certain cancers. Such genes we call it as proto-oncogenes, and such kind of genes containing person we say they have pre-malignant tumors. What is the treatment for the cancer? Treatment of cancer. The common approaches for treatment of cancer are surgery, radiation therapy, and immunotherapy. In radiotherapy, tumor cells are irradiated lethally, taking proper care of the normal tissues surrounding the tumor mass. Several chemotherapeutic drugs are used to kill cancerous cells. Some of these are specific for particular tumors. Majority of drugs have side effects like hair loss, alopecia we say, hair loss, and anemia, reducing the number of RBC. Most cancers are treated by combination of surgery, radiotherapy, and also chemotherapy. Tumor cells have been shown to avoid detection and destruction by immune system. Therefore, the patients are given substances like biological response modifiers such as alpha interferons. Biological response modifiers like alpha interferons, which activate their immune system and help in destroying the tumor. So this is a brief explanation of the cancer biology here. Thank you. SRIKANTH.